cryo-electron tomography, which is a very popular technique now. It was used to on the structural study of macromolecular complexes as well as cellular structure, particularly in their native environment. So this paper I give you is actually a review article. It will particularly focus on how you prepare a sample for cryo-electron tomography or cryo-ET. So here it contains several topics. First, it will show you how to prepare a cryo sample using the punch freezing. And then, because the sample is too thick, we need to thin down the sample. We'll use the focused ion beam or FIB to create this very thin sample called lamina. And then you will describe how to use the fluorescence labeled protein as a marker to guide the sample preparation as well as the data process. And in the end, I will also give you 2 or 3 slides to briefly talk about how the cryo-ET experiment is conducted and how we process the image. But first of all, let's look at this cryo-electron tomography, cryo-ET. We can divide this into 3 parts: cryo, electron, and tomography. Here, we would like to ask ourselves a question. What is the advantage of using electron? And the second, why does the sample have to be cryoprotected? And lastly, how does the entire cryo-ET experiment is conducted? So to answer the first question, here, electron. Why is the advantage of using electron beam? We have to go back to understand how does the resolution is determined in any given microscope. So you may know some microscope has better resolution, can see much more detail, but some are not. What factor determine that? So to answer this question, let's go back to the definition of the resolution. So the resolution is the minimal distance at which 2 points can be clearly resolved. So if you can see here in your left hand, there are 2 points or 2 peaks. It is very clear to you there are 2 points, so we will call it resolved. But if these 2 points get very closer and closer, in the end it will overlap 2 points together. To your eye, maybe just 1 point, so we will say it's not resolved. Somewhere in between, there's a point, a place. You can just see, well, it's very close, but you can still tell there are 2 points. We'll call it just resolved, or technically it's called Rayleigh limit. So the distance of this case is much smaller than this one. So this is the resolution limit for this particular microscope. So mathematically, it can be written down like this, so-called Rayleigh equation. We look at this equation. The resolution here, delta, is equal to 0.61 and lambda. Lambda is a wavelength over mu psi theta. This one, the bottom one, is a constant number for a given microscope. So we temporarily ignore it now. If you look at this, you can see that for a microscope, your resolution is correlated to the wavelength of the incident beam. Is that okay? Nearly your resolution is about half, 0.6 or half of the wavelength. Okay, once you get this idea, let's go back to see the entire spectrum. From the top, the smallest wavelength, and the bottom, the longest wavelength. So from the top, you can check on your screen that I already uploaded a slide. From the top, we'll have electron, gamma ray, X-ray, UV light, visible light, IR, and so on. Let's take a look at the visible light. You will have a wavelength from 400 to 700 nanometer. If you consider what we talked about in the previous slide, the best for a good visible microscope, the resolution is about 200 nanometer to 750 nanometer. So the light microscope resolution 300 or so. That's a limitation for the microscope in theoretical way. So then, what scale do you need to see, let's say, an atom, a peptide bond, a helix, a protein, or a cell? So before you start your experiment, you have to know which microscope to use. What scale do you need to see an atom? Here, I'll show you the peptide bond structure I use in undergraduate course. So you can see here, the peptide bond here is 1.3 angstrom. So pretty much all the chemical bond is the same, about 1 to 2 angstrom covalent bond. So what wavelengths do you need? Twice, about 3 angstrom wavelengths in order to see the atom or chemical peptide bond. Is that clear so far? Yeah. So 3 angstrom wavelengths, what they are? X-ray. X-ray is here, angstrom range. That's why we usually use X-ray crystallography to determine the protein structure. But look at this, electron. Electron has much more potential to see the finer detail in theory so far. Theoretically, it can see, had created much smaller wavelengths. You can see the much detail, finer detail. Okay, you got the first concept. Then let's see why do we need to do the cryo? Why not just room temperature? Why does your sample have to be protected in cryo temperature? Let's say if we have a microscope here, on the top we will have electron gun. And electron will emit from its top and go until it reaches the sample, usually in the middle of the electron microscope. It will penetrate your sample until it reaches the detector. So the electron will move from top all the way to the bottom. It moves straightly to the bottom until it can be detected by the electron detector. But if there is a gas molecule inside your microscope, it can scatter the electron. So your electron will no longer move directly, straightforward. It will change its direction. Therefore, our microscope usually keep in a high vacuum to make sure the movement of the electron. That's the first. And also, once electron reach your sample in the middle of a microscope, it can transfer its energy to the surrounding. It will be your sample. This energy can ionize atom, and sometimes it even break the chemical bond. So this is called radiation damage, or you can just think as a heat damage. You actually heat up your sample and eventually destroy it. So our biological sample will not be able to tolerate these two, high vacuum and high intensity electron. That's why scientists find a way to resolve it. It's called cryoprotection, which means keep your sample at so-called cryogenic temperature. This is minus 180 degrees Celsius temperature. And this cooling process has to be done very quick in order to prevent the formation of ice crystal. Because the ice crystal can destroy your structure. It's very bad things. So cooling, later on we'll have movie, but this whole process ultra speed. So you will keep your sample in so-called vitreous ice condition. This vitreous ice is somewhere between water and ice. It's more like gel-like structure. It will preserve your sample in nearly native water-like condition, but not completely ice. So because this process is highly related to our next hour's group presentation, so we take a look to see how this is done. So the key point for this so-called conventional punch freezing is you have to control the whole process very quick to prevent the formation of ice cubing. Okay, let's start to take a look.
In preparation for plunge freezing, a brass cup is inserted into the center of an insulated bowl, followed by the grid box pedestal, the spider unit, and a float. The assembly is filled with liquid nitrogen. After the temperature equilibrates, the brass cup is filled with a cryogen, typically liquid ethane. Filter paper is attached to the blotting pads, and the tweezers are mounted onto the plunge freezer. The cryogen bowl is placed onto the plunge freezer, which is then raised against the sample loading chamber. A few microliters of sample are applied to the grid, after which the grid is blotted by the blotting pads and immediately plunge frozen into the ethane. Finally, the grid is carefully transferred from the ethane to the grid box.
So you can see the whole process. Well, it's okay, but it really depends if you are handy or not. It will take a few minutes. Quickest, still need take a few minutes to finish one grid. So you won't be able to deal with millisecond reaction if you really want to do time resolve experiment. So later on, next hour, we'll have a new device talking about the newly way to freeze your sample. Alright, so now you know these two, why electron, why cryo. Now let's look at what is cryo-EM tomography. So go back to the tomography, this word. The first tomo means section, a slide, a few slides. This tomography is already widely used in many medical and scientific fields. As you can see here, there's called PET, positron emission tomography, also the MRI and the CT, they are pretty much the same. The human being is lying in the chamber, and there's a detector circulating around this object in order to take a lot of 2D image. So once it collects all the 2D slide, it will combine to all the slide and generate the 3D object information. So from the slide, you can see many slides, then you will have this 2D model or 3D model or 3D reconstruction. So this is the word tomography means. So now we go to the cryotomography, but go back one step before digging into the cryotomography. We talk about the cryo-EM. Currently, cryo-EM have two major types. Single particle cryo-EM analysis, you must heard it a lot. This is the first type. And the second type is called cryotomography. So far, single particle cryo-EM analysis is still the mainstream in the field. But this one is catching up. It has a lot of potential and a lot of advantage we'll talk about later. But for single particle cryo-EM, like the word said, you need a single particle, which usually means purified the micromolecule complex also. So sample have to purify in order to obtain this homogeneous single particle. But what if your sample cannot be purified? Then you will have to use cryotomography. This is usually used on some unique sample, and you want to understand how this, let's say, nuclear probe, how this organelle deal with its neighboring environment. How does it interact with other organelle? In this case, you will have to use cryotomography. You cannot just purify your subject. So that's a major two difference. One has to purify, and this one we don't purify it. We study how they react to their neighboring environment. So this sample exists in their native context. Okay? So a quick movie shows cryotomography, and in this case, we freeze a cell. We want to study one particular cell. So we have a lot of cell, then we freeze it, use punch freezing. So we have a lot of cell on this is EN green. So next we put this sample inside the microscope and we tilt it to collect all the different angles. So this, a lot of 2D slide is called tilt series. So you want as many angles as enough in order to obtain all the spatial information. And then all this one will be back projected to create a 3D volume. And this 3D volume information is called tomogram. So from this tomogram, we'll be able to extract information and build a 3D reconstruction. That's the basic workflow of the cryotomography. So now you look very fancy, but is the cryotomography the right tool for you? It really depends on your project. There's nothing 100% good or nothing 100% bad. If you look at here, we will have from the microscope, light microscope, cryotomography, single particle cryo-EM, X-ray crystallography, MNR. So this different resolution this tool can reach, and also what they can see. From atom, protein, virus, and the cell. They all have different application, different optical range. But one thing you probably need to notice is that for these three tools, MNR, X-ray, and single particle cryo-EM analysis, they have to deal with purified sample, recombinant protein, or so on. So these three samples need to be purified. But for these two, we usually don't purify sample like I mentioned before. But cryotomography is the one can bridge atomic resolution to micrometer. So how big is a cell? Let's say a human cell, how big is it? How big is your protein? So once you understand your target of interest, you will know what to choose. Okay. Now let's say at this point, you think you have your sample, however, a cell of something frozen by punch freezing. You think you are ready to collect, put it into a microscope and collect it, which is not true. Now, once you have your frozen sample, you encounter the first limitation of cryo-ET, which is sample thickness. Because all this is because our electron cannot penetrate sample thicker than, well, here write 500 nanometer. In reality, actually 200 or 300 nanometer. If electron cannot pass through, penetrate your sample, it cannot reach the detector. This is related to the electron scattering characteristic. We don't talk about this. But anyway, your sample has to be very thin, but how thick is your sample? Let's say, if you are working on a human cell, you may know the diameter, like a couple, 20 micrometer, but what is the thickness of your sample? In most cases, eukaryotic cells are much thicker than this so-called 500 nanometer. What should we do? We make it thinner, just thin enough to see what we want to see. So there are two ways to trim your sample. One is called cryosection, just cut it, use knife, diamond knife also, but we don't really use that. It's very laborious, it's difficult to control. Current most popular way is using ion beam to cut it. So in cryo-ET, we use this cryo-focused ion beam or cryo-fit mining technique. So this will thin your sample when your sample are maintained in their freezing status. So cryo-mining this technique. Let's look at another movie to let you have better idea how this experiment is done.
Cryo-electron tomography provides 3D snapshots of cells and visualizes protein complexes at molecular resolution. The workflow begins with cells, which are prepared by routine cell culture methods on electron microscopy grids. Cells are preserved in their natural hydrated state by rapid freezing in a vitrobot. Vitrification cools the sample so fast that water molecules do not have time to crystallize, avoiding damage caused by the formation of crystals at slower freezing rates. To achieve the rapid cooldown of the sample, a thin film of liquid is required. For this purpose, the vitrobot automatically removes excess buffer by blotting the grids. Immediately after, the cells are automatically immersed in liquid ethane at minus 180 degrees Celsius. The flash-frozen cells are now vitrified, and the EM grid is transferred to the cryofib. The Aquilose cryofib is a dedicated cryo dual-beam microscope. This instrument is used to produce thin, electron-transparent samples from the cells, which can be imaged later by cryotomography in the cryos. For preparation, the frozen EM grid is mounted on a cryo holder and loaded onto the cryo stage. The Aquilose system ensures cells are kept vitreous at cryo temperature during loading and fib milling. Next, the stage is rotated and a gas injection system is used to apply a platinum layer, which protects the sample from ion beam erosion. Inside the cryo dual beam microscope, the electron beam is used for imaging the sample, and the ion beam ensures precise removal of material from vitrified cells. To excise a region of interest from a vitrified single cell or a high-pressure frozen sample, the areas to be removed are first marked. The ion beam is then used to precisely remove frozen cellular material. In the next step, the region of interest is then secured to the cold needle of the EasyLift system and afterwards cut free with the ion beam. The cutout sample is then transferred to a second EM grid inside the Aquilose. This grid contains small side posts where the lamella is attached. After attachment, the needle is cut free. For the last step, the bulk lamella sample is thinned down using the ion beam until it is about 200 nanometers thin. The frozen cell sample is now thin enough so that electrons can pass through during electron tomography acquisition. In the last step, the in-chamber sputter coating system is used to apply a thin conductive layer of platinum on top of the lamella sample to prevent that it charges up during 3D imaging in the TEM. The sample is now transferred to the Cryos TEM system. The Cryos is the leading cryo-EM platform and a 300 kilovolt transmission electron microscope. It allows loading of multiple grids using an automated sample loading system. During cryo-electron tomography, an image series is acquired as the fib-prepared lamella is tilted incrementally. Finally, the images are combined by computer into a 3D tomogram. The tomogram is a snapshot of the preserved hydrated cell interior, where protein structures can be explored in their native environments.
So this movie is from the Thermo Fisher, which they want to sell the machine. So they make it look very colorful and very easy. But let's take a look at what really happened in reality when you cut the cell. So first of all, just to remind you, when you do this cryo-fib milling, you have two beams. One is electron beam to observe the sample. One is ion beam to cut the sample. So you can use ion beam to use electron beam to see where to cut, and they both can be done simultaneously. So you will need to choose which cell to cut and which part to left in order to produce this very thin sample called lamella. Okay, let's take a look. So first you have a grid, and you have a tiny cell here. These little dots are the cell. From the side view, you have like a little hill. So in this case, you choose this one and apply ion beam to cut the top and cut the bottom. Right now, we remove the top of the cell. So notice this. Now everything is grayish. There's no particular cover. So if you want to work on the nucleus, where is the nucleus? Where is the mitochondria? Right? Now we continue to cut it. Speed it up a little bit. So now you generate this one lamina. You can have several lamina for you to observe in the microscope. Here, the figure in the paper on the same, it shows that some cells grow on the grid, and you apply ion beam to make the cell thinner, and also observe using the electron beam. So you have sample in this case about 200 nanometers, very thin lamina. But like I said, before you start your mining experiment, you will have to know where to cut. If you consider this, you will have no idea where it is, where you want to see, where is your target of interest. That's why the second major limitation is it's difficult to find a specific area within this grayscale value of your cell. So here comes the second one, the second part, correlative light and the electron microscope, we call CLIM. It will use fluorescence label, usually protein, as the marker to guide in order to see where the sample, your target of interest is. So you will combine both light microscope and electron microscope. You basically use the fluorescence image from light microscope and apply it to electron microscope during the cutting. Let's see one case provided by this paper. So here we have sample, but here this one is microphage that grow on the grid, and this cell will infect with the bacteria. So here the nuclei from the cell is labeled in blue. It's a blue fluorescence protein, and also bacteria is labeled in green. That's very easy. You just express GFP in the bacteria, you will get a green signal. So here you can have green and blue. You will have some sense to where is your sample is. This is a nice one. So here again, it has a microphage infected with E. coli. E. coli is shown in the green. So in this case, they want to see the E. coli within the microphage. So you just cut it, but keep the green signal within your lamina. Then in this case, the red circle here also indicates some, in this case, they added some gold particle to help alignment. But just focus on this green GFP signal that indicate where your feature is. So finally, this one is another example. They want to see this peroxisome membrane protein inside the cell wall, whatever. So this peroxisome membrane protein was labeled fused to GFP, and in order to locate this vesicle in the yeast. And also they somehow label the cell wall in blue, so you will have cell wall and this GFP. So they start to do the preparation, and then freeze the sample, do the mining, and collect the whole data set. So this is a tomography after data collection. If you can see here, they have a very strong green signal here, indicate the position of peroxisome vesicles here. So later on, they can build the structure based on this indication. So far, this part will just focus on how you prepare your sample for the cryotomography. But when it's difficult to see it, you can apply the clean technique on your sample. But later, to let you have a better idea, although it's not covered on this topic, and this paper will just use a few slides to let you see what will happen next. When you have your sample ready, you can just insert into your microscope and start to till it. You should till as much as possible in order to extract all the spatial information you can get. So all this, each slide will put together. We'll have many slides like this, from the plus 60 degree to minus 60 degree. All these slides have to align because they have, during the tilling, they have like a sliding movement. So this alignment, once they put it together, and then you can build a 3D volume tomograph. So this is a nice cartoon to see how, to let you see how this slide will build together.
During data collection, an array of images called a tilt series is collected at different sample orientations. The sample is tilted gradually, and a low-dose image is collected at each step until an angle of 60 degrees is reached. The sample is returned to its starting position, and images at other tilts are collected until negative 60 degrees is reached. A tilt series records 3D information from different views from a single sample. Near perfect alignment of images in the tilt series is crucial to obtain accurate, high-resolution 3D reconstructions. Horizontal, vertical, and rotational whole image shifts and smaller distortions are corrected during alignment and iterative refinement steps.
So you can see here, you have each one is one angle. So you have to combine all the different tilt series together and partially align it based on what reference you can get, and put everything together to get this 3D information. So in the end, all this one, you can get a 3D reconstruction bed, which will have what your sample will look like. Okay, my part will pretty much finish here, but something worth to notice that right now, first you know why the electron microscope is getting more popular and why the sample really have to be cryoprotection as a key factor. But so far, although the electron has, because of its smaller wavelengths, it has very good potential. So far we cannot build a perfect microscope. That's one reason the resolution is still about angstrom. It's not like 0.2, 0.01 angstrom. There's still some restriction. And then another key factor is how do you prepare your cryo sample. So far we can only use this punch freezing. That's the best we can commercially have now, but there are many groups trying to develop a more advanced device in order to see more detail of the structure. So later on, next hour, we will have a group to present this super cool time-resolved device to freeze sample. Okay, I guess that's it. Thank you.